Greetings, humans. All right, we are still in chapter 3, and now it's time to talk about neurons. The neuron is the smallest unit of cognition, let's say, the smallest biological entity we can find that seems to have implications for cognition, for thinking. So the brain is basically made out of 3 kinds of cells. You have astrocytes, glial cells, and neurons. Astrocyte means star-shaped cell, and these really seem to just give us the loose skeletal structure of the brain. To the extent that the brain has structure, like if you poke a hole in someone's skull, then the brain will try to swell out of that hole. Maybe you've seen police procedurals and stuff, and they're doing an autopsy, take off the top of the skull and reach in and just handle the brain. You can't handle a brain without extensive preparation of the neural material. It's kind of like quick set Jello. It's sloppy, it's messy. It really needs the shape of the skull to retain its shape. So loose, absolutely loose skeletal structure of the brain, that's the astrocytes. And then the other cells in the brain kind of lodge between the dendrites and branches of the astrocytes. Okay, glial cells seem to be sort of the sanitation engineers, the waste collectors of the brain. Like everything that lives eats and everything that eats poops, okay? So neurons, brain cells are in there doing their thing. They're respiring, they're putting oxygen and sugar into energy and doing their function. And so as they do that, they create a waste product. And the glial cells are there kind of to get the waste products out and some other sort of maintenance functions. They're the groundskeepers enabling the rest of the neural structure of the brain to do what it does. And you have neurons, and these are the parts that really seem to be doing thought. Okay. So parts of a neuron, you have your cell body, and it has all the same stuff in it that all the cells have, right? It has all 46 chromosomes, it has the mitochondria, the power plant, has whatever it is, the thing that's the wavy lines. What is this, biology class? I don't know what's in there. And then the cell body has an axon that trails away. The axon is the sort of the cable that connects it to other neurons. Each neuron might touch up to 10,000 other neurons. I'm thinking about a sidetrack. Maybe I'll remember the sidetrack in a second. Dendrites, so all around the outside, so here's your cell body, and then all around it are going to be these branches that reach off, and dendrite just means branches, okay? So that's where the terminal buds, the end of the axons of other neurons are going to touch. This is where neurons touch each other, okay? So at the end of the axon, let's reorganize this a little bit. At the end of the axon, you'll find the terminal buds. If you like, you can call those terminal buttons. In my books that I read when I was an undergrad, it certainly was called terminal buttons. And we were encouraged to say terminal boutons because the dude who discovered all this stuff for the first time was French. So you can French it up if you want to and say bouton instead of buttons. No judgment here. Okay, and then we have synapses, and the synapse is where those terminal buds reach and touch the dendrites of the next neuron in line. So you have neuron, axon, dendrite, cell body, axon, terminal buttons, right? Dendrites, cell body, like that. Doesn't exactly quite touch them. The brain is an electrochemical system. The axon is the electro end. The synapse is the chemical end. The terminal bud or button never actually quite touches the dendrite. There's a tiny microscopic gap between them, and that gap is closed by neurotransmitters, which we'll get to in a second. Okay, how does this work? When it's not firing, The axon is gathering sodium and potassium ions up at the start end next to the cell body. What it wants is a ton of extra electrons. It's gathering atoms that have too many electrons, so they're unstable. Okay? And then when it's triggered, when it gets enough charge through its dendrites, it releases all those extra electrons, and they run down the axon. They get into those terminal buttons and open what's called synaptic vesicles, which sounds dirty but isn't. Vesicle is merely a container. The containers contain neurotransmitters, right? So if enough electrons make it to the end, they rupture these vesicles, and then neurotransmitter wafts across the synaptic cleft. So here's your sending end, here's your receiving end, all right? And then over here, you're going to have a bunch of receptor sites for that neurotransmitter. Neurotransmitter crosses the cleft. If enough of the receptor sites at this end are filled, then that's what's triggering the next electron rush from the next neuron in line. Okay, fantastic. We got that straight. Good. Encoding. So one neuron is essentially useless to understand. Again, this is not a biology class. Why are we talking about this? Understanding a neuron gives you no understanding whatsoever about human consciousness, thought, or decision making, or anything. It's useless. Information is not encoded in one neuron. Information is encoded in networks of neurons. Neural networks are where the action is today. So if you learn nothing else today, what you need to know is that neurons that fire together wire together. Say it with me. Neurons that fire together wire together. Congratulations, you've learned all you really need to know as a psychologist from the biopsych chapter. Wonderful. How's it work? Well, let's say you're 1 year old and you're out in the perambulator. Americans would call it a stroller. You're out in the stroller and your mom's taking you to the shops to get milk because babies drink milk, and you see a dog. You've never seen a dog before in your life. You have no idea. So you point at the thing and you go, "What's that?" And your mom goes, "That's a dog." All right. So as you're having this experience, some neurons in your brain are firing, and they're firing together and they're wiring together. Each time the receiving end of the synapse receives neurotransmitter, it makes more receptor sites for that neurotransmitter, making it more likely in the future you can fill enough of those sites, they can catch enough of their neurotransmitter that we can keep that connection going, right? So a neural network fires and it encodes the experience of dog, right? So from now on, whenever you see a dog, those neurons will fire. And whenever those neurons fire, you will see a dog, right? So right now, you can probably close your eyes and relax. If you haven't already, listen to me, right? Close your eyes and relax a little bit and imagine in your head a dog. It has dogness and greasy, wiry hair and a wet nose, and it loves you unconditionally, and it smells funny, right? You practically smell the dog. Okay, fantastic. So the next week you're out because you drank all the milk, selfish baby. So you're on your way back to the shops for more milk or something, right? In the stroller. And this time you see a cat. But as far as you're concerned, you know, it has 4 paws and a fluffy tail and whiskers and a little nose and sticking up ears. So you point at the cat, thinking you're smart. You go, "Dog," right? And your ma goes, "No, you idiot, that's a cat." Okay, so now your brain has a choice. I mean, choice metaphorically. It's not actually making a decision the way that you're thinking, but it is a decision point. You can choose assimilation or accommodation, right? So imagine you have this network of neurons for dogs, and you could assimilate this new information. You could put cat into the mix as a kind of dog, right? How many kinds of dogs are there? You're already doing this, like Doberman Pinschers, and giant Alaskan Malamutes, and Huskies, and Poodles, and Beagles, and not Chihuahuas. They're not dogs. They're like a very specific breed of rat. Do you get the picture? They all fit in this neural network somewhere. You could just add one more and go, well, some kinds of dogs are cats. Okay? Or you could do accommodation. And in accommodation, you'd start a whole new neural network for brand new, fresh information. So this is college, and if you're in my class, you're a grown-up type human, you're an adult, and I don't care what your bio age is, you're doing assimilation. I'm encouraging assimilation over accommodation. Why? Because by age 5, you have 100 billion or so neurons in your brain. But neurons take up space and they're expensive to maintain. So the ones that are not co-opted into neural networks get pruned away. It's called developmental pruning. So that by age 30, you're left with only 40 billion neurons in your brain, right? Synapses take up space, and synapses are really where information is encoded. So neurogenesis slows down and almost stops. Synaptogenesis takes over, working those neurons into neural networks. The thing is that as an adult, it's much harder for you to create whole new neural networks for fresh information. It's much easier for you because you have so many hooks already in your cloakroom. You just hang codes on the hooks already present in that cloakroom. Do you have cloakrooms in America? Look it up. Good. So you're probably an assimilator, and that means as you're trying to learn stuff, you should try to come up with metaphors for it, to think of it in terms of stuff you already know. Will be a much easier proposition for the adult. Okay, so that's encoding. Just remember this, honestly. Neurons that fire together wire together. If you can remember that, you're doing pretty good for the rest of the chapter. It's going to come up almost every chapter hereafter. Okay. Neurotransmitters, there's a nice list in the book. It's like a table of neurotransmitters and what they do. And honestly, I don't care about it. Like, why are we talking about this? This is psychology. Biology really doesn't have anything to do with it. You can take a biology class and get a much clearer, better informed, solid perspective on this stuff. If you care, I encourage you to do that. For now, sort of bookmark somehow the page that has that list of neurotransmitters in case they pop up on test questions in the mastery quiz. You know, they're open book, use the book. I really don't want you to memorize it. Neurotransmitters can do lots of different stuff, including crossing the synapse is one function. Serotonin is implicated in crossing the synaptic cleft. Epinephrine and norepinephrine are also implicated in doing that direct fire signal thing. But they also can bind along the axon and in other places and raise or lower the firing threshold for neurons, okay? So if we raise the firing threshold, that means that we need more charge to crack open those synaptic vesicles to get the neurotransmitter. And if we lower the firing threshold, we need less charge. You know, it's more of a hair trigger to get those vesicles open, right? So a higher firing threshold means the brain is slowing down. And a lower firing threshold means that it's speeding up. And a lot of the action of neurotransmitters is about this, making it harder or easier for brains to fire. So that's the two basically more or less things that neurotransmitters do, but they do it in different parts of your brain and for different reasons at different times, okay? So let's talk about side effects. Parsimony means being unwilling to spend unless spending is really necessary. Heard of someone who's tight-fisted? They hold on to their money really tightly. They keep a tight grip on the purse strings, right? That's parsimony, okay? So why would your body make kinds of molecule when it can make one kind of molecule and get that molecule to do different things? Exactly. Neurotransmitters are found not just throughout the brain doing different functions, but throughout the body doing different functions. Take dopamine. Dopamine is implicated in digestion. You have dopamine in your gut and in your liver, and they're regulating digestive processes. You also have dopamine in your brain. Okay, but you have dopamine in different parts of your brain doing different things. Dopamine in your motor cortex, for example, is damping down unnecessary impulses. In other words, it's raising the firing threshold for neural networks in the motor cortex. What does that mean? It means if you have a dopamine shortage, you start to get random movement. Dopamine's not there to damp down the firing, and so neurons are firing kind of at random, and then you'll get the sort of Parkinsonian tremors associated with Parkinson's disease and with antipsychotic medication. When it's in response to medication, that's called tardive dyskinesia, right? A side effect of antipsychotic medication. So you have dopamine in different parts of your brain. It seems to regulate attention. When it's sort of in the midbrain, it's about saying pay attention to this, but not that. What are the symptoms of psychosis or schizophrenia? Hallucinations. So everyone has an inner voice, right? Your inner voice is talking right now, but you recognize that your inner voice is coming from inside, and you might kind of ignore it and pay attention to voices on the outside and weight them and give them priority accordingly. But the psychotic person has lost the ability to do that. The dopamine shortage in the midbrain is making it so they're attending to inner stimuli as though they were external stimuli, right? Okay, so what might be a cure for that? I'm going to give you dopamine. Sounds good. The thing is, I give you dopamine in a pill, and you swallow the pill, and the dopamine goes all over your body, right? The bloodstream doesn't carry it straight to the midbrain. The dopamine carries it all over the body and all through the brain. So you might have metabolic side effects. In fact, the newer classes, the atypical antipsychotics, you can more or less expect people to get fluffy and to have a risk of heart arrhythmia, right? Because it's going everywhere in the body. But also, what happens is excess dopamine burns out the dopamine receptors in the motor cortex, causing effectively a dopamine shortage. The dopamine's there, it just doesn't have anywhere to land. Your motor cortex has become immune to it, and so it's acting like it's not there, right? Which means the dopamine's not regulating those Parkinsonian movements. So a person taking, especially an older antipsychotic, can expect the tongue rolling and the foot shuffling and the back and forth kinds of movements associated with Parkinson's disease. When you have tardive dyskinesia, it's permanent. There's no cure for it. I can't regenerate the dopamine receptors in your brain. Right? So that's side effects. That's why we get side effects, right? Almost every drug you take, it's going to have a side effect profile, and you read them and you accept whatever side effects as a treatment for the thing that you have. Because I can't target where in your brain we're putting those neurotransmitters or neurotransmitter analogs. What else is important here? Yeah, I'm having a thought, and maybe we'll get to it. Okay. So what? So what do we do with all this? What do we do with all this information? Here's a thought I'm having. It is agonists and antagonists. Let's do this first. It's important, and then we'll get to the so what here presently. Okay. Good. So imagine a key and a lock. Neurotransmitters are the keys, the receptor sites are the locks, right? Okay, so maybe the chemicals I'm putting in your brain aren't neurotransmitters, but they're like neurotransmitters. Their molecular shape is close enough for government work. Okay, so maybe I want to give you the effect. So caffeine, for example, is somewhere in the acetylcholine matrix. Um, so I want caffeine to fit in the receptor site for one of the acetylcholine derivatives, and I want it to turn the key. I want it to give you the effect as though you have that neurotransmitter. Okay, so caffeine looks enough like, it's the key shaped enough like the neurotransmitter that it fits in the lock, and it turns the key and it produces the effect that neurotransmitter would have had. That's an agonist. Okay, but sometimes these analogs are antagonists. They fit in the lock, but they don't turn. In other words, they're just blocking that receptor site. If I were to give you a serotonin antagonist, then it would bond in the receptor sites for serotonin, but then serotonin would just bounce, right? And you wouldn't get the effect of that serotonin because it can't fill enough of the receptor sites. That's an antagonist. How do you remember that? Maybe you've had a bad breakup with a boyfriend or something, and so you still have his car keys, and you really hate him, and you want to make sure he knows you really hate him on the way out. So you take the car key and you put it in the car door, and then you kick it really hard and bust it off, right? So now the key's left in the door, and there's no handle to turn it with, and he has to see a locksmith and spend $400, to have his key. That would be an antagonistic thing to do, wouldn't it? Right? So that's an antagonist. It goes in the lock, doesn't turn, just blocks it. Okay. So what are we to do with all of this information? The biopsychology people, as we said earlier, they really want to link cause and effect in maybe some undue ways. So here goes the serotonin theory of depression. Let's go back up to synapses a little bit. You're sitting and trying to have a thought, right? And this neuron activates, and it reaches out and touches maybe 10,000 other neurons, wafting its neurotransmitters across the various synaptic clefts. Well, it's not as simple as there's the sending end and the receiving end. The synaptic vesicles rupture, and then those neurotransmitters kind of waft into the gap. Like you're throwing balloons, though, they don't shoot across like a t-shirt cap at a concert, they waft. And then because of the electromagnetic force, they're attracted to the receptor sites. The thing is, the synaptic vesicles basically are also receptor sites. The difference between the vesicle and the receptor site is so minor, they act the same way, and once those neurotransmitters get into the gap, now it's kind of a tug of war. These guys are reaching out and trying to grab all the tasty neurotransmitters. These guys are too. The electromagnetic force is pulling them both ways, and so it's kind of chance whether enough make it to this side, and that's why every time this guy gets neurotransmitters, it makes more receptor sites, so it win that tug of war more and more. So the serotonin theory of depression is that reuptake is too efficient. What's happening is the vesicles are winning that tug of war more often than they should. They're grabbing back their neurotransmitter load. They're not closing the circuit. And so your thoughts are slowing down. It's getting harder and harder to complete a thought. That results in depressed mood and memory problems and appetite problems and so on. All stuff linked to serotonin. Cool. So what do we have? We have a drug, selective serotonin reuptake inhibitors, or SSRIs. Okay. Those are antagonists. And they bond differentially. They bond only with the synaptic vesicles, not with the receptor sites. So they're wafting around, they get in the bloodstream, it takes a couple weeks to build up, to be in the brain enough, right? And they're kind of wafting around in the synaptic cleft. The vesicles rupture, they send out the neurotransmitter, and then they try and snatch everything back, but they're snatching up a bunch of this SSRI along with the neurotransmitter. So now there's enough neurotransmitter left to make it across the cleft, and you start to complete more thoughts, and your brain speeds up, and so on. Fantastic. Miracles of modern science. What if, though? What if the only evidence for the serotonin theory of depression came from the for-profit pharma companies producing antidepressants, making tens of billions of dollars a year in America alone on antidepressants alone? And what if that were true? Because that's the case, right? And remember the correlation causation thing. The experimental evidence here isn't very good. How do we do this? Well, first of all, we're going to conduct our studies in Denmark and Sweden and Switzerland and stuff. Why? Well, because we get to escape oversight, and we get to bury the results that we don't like. So what we're going to do is we're going to recruit 1,000 people into a population. We need 1,000 people who experience depression, and we're going to give half of them the medication. We're going to give half of them a placebo, and we're going to see who's more better at the end of the study. Hey, I used more better correctly, because everyone's better at the end of the study, right? Depression tends to go away with time, and we give it a year or 6 months or something. We measure depression before, depression after. Both populations are better on average, but who's more better? The thing is that I don't stop by getting 1,000 people and then putting them into 2 roughly even groups with random assignment. No, I break them down into groups of 50, or better still, 25. How many groups of 25 can I get out of 100? Out of 1,000, it's 4 per 100, so 40 groups of 25 each. That's what I'm going to wind up with. And in each group, I'm going to give half of them the placebo and half of them the active ingredient. Why? Well, if the medication does nothing, if it's as good as the placebo, then in some of those groups, more people will get more better taking the medication than the placebo by chance. Correspondingly, in some of the groups, people taking the placebo will get a better effect than people taking the active ingredient. And in most of the groups, there won't be any difference because the medication does nothing. Then I can publish the 2 or 3 of those studies of 25 individuals that show that the medication was effective and had low or no side effect profile, and bury the remainder, bury the other 37, just don't publish them. Okay? This is called positivity bias. Journals only want to publish studies that show something true. They don't want to publish studies that show something false. It's not sexy. You can't sell it. It doesn't have a good headline. It's not making anybody famous. But negative results are results. They're important results. And in this case, what we have is a whole field of medical literature telling us medication is effective, whereas if we could see all of the data, we'd find that it's not. It just isn't. So are these corrupt drug studies sufficient evidence to say that depression is a serious medical condition caused by a chemical imbalance? That's just company propaganda. And honestly, the 2010 Journal of the Medical Association, JAMA, published a meta-analysis where they didn't even go and find all the missing studies. They just looked at the studies that had been done, lumped all the data into one pool to get more statistical power. And they find that, except for the most serious cases of depression, nothing beats placebo. Okay, and if nothing beats placebo, how can we say that depression is caused by a chemical imbalance? What other possible factors could lead people to experience depression? How about poverty? How about relative poverty? If you're working a menial service job and you look at other people working less hard for a lot more money, taking vacations, and you're eating at McDonald's and they're eating at some fancy place, posting it to Instagram, living their best life and whatever, you might start to feel depression. Do you go to work every day and do just menial service work where you're never going to get ahead, right? The company has no loyalty to you. You make minimum wage or less. They work you 35.5 hours, because if they work you 36, they'd have to give you benefits. So you're part-time, no benefits, no insurance, no retirement plan, no possibility to move up in the organization, and easily replaceable, right? If you quit in protest, there's people lining up behind you to take the crappy low-wage part. Why wouldn't you be depressed? Maybe you're a person of color in a racist country. It's pretty depressing. Maybe you're a woman in a sexist country. You're not lucky enough to benefit from the patriarchy. That's pretty depressing. Life is depressing. And some people respond relatively well to the depressing aspects of life, some relatively poorly, but life is depressing. The brain encodes information. Your brain knows whether it should be depressed or not. When we give someone a course of antidepressants, if they work on average for about 9 months, and then we need to trick the brain with a new medication because we haven't done anything to change your circumstances. Is this bad? Possibly. Possibly. I don't want to say for sure. But antidepressants have what's called a black box warning on them. That means when you get your prescription, it comes with a tiny little square of paper like this. You unfold it like a map, and it's gigantic and all in 4-point font, and you need a magnifying glass to read it. Okay? But there's one warning with a black box drawn around it to draw your attention. That's why it's called the black box warning. It says, this medication may cause suicidal or homicidal thoughts. They don't know for sure. They're really cagey about saying that antidepressants cause homicidal ideation. That would be weird, right? A med that causes a specific ideation. But maybe it's this. Imagine you're a white cishet guy growing up in rural America, in flyover country, in the Rust Belt, and used to have one kind of life. In that kind of life, the church was the center of the community. Everybody went there. It's where you met babies for the first time because you're born and you get christened there. It's where people get married. It's where you met your person, and all the town business is done there. AA meets in the basement. Bingo on Friday nights. If someone dies, it's the church that organizes people to go and take casseroles on given nights and get people through it. Right? Church, absolutely the center of the community. And there was a manufacturing plant and everybody worked there. They complained about it, but it was the life of the community. All that's gone now. Manufacturing is gone. The center is hollowed out. People look at religion like it's just a weird superstition, a conspiracy theory. All of the movies are about how rural people are cannibals who can't be trusted. Inbred freaks who will eat you if your car breaks down. It's gone. That way of life is gone. The church is still there, but only the old folks go, and the jobs are gone, and there's nothing to do. Massive meth problem. What's the other thing? Opioid problem really hits the suburbs and farm country the hardest because there's nothing else to do. You're depressed. In fact, you're mad about it. So you suck it up and go to the doctor, which itself is really an act of courage and determination. You go to the doctor and you tell the doctor all these symptoms, and they go, oh, you don't have a problem. You have a chemical imbalance, right? Here's some drugs. Take the drugs and you will feel better. They've totally steamrollered over all of your thoughts and your feelings and your concerns, right? They've totally invalidated everything happening in your head. They're saying you're mentally ill for feeling this way. The one person who's supposed to listen to you and take your concerns seriously is invalidated. Why would it be a surprise that in a small, very small percentage of these cases, people pick up a gun and go to school or go to work? Why would it be a surprise that in a slightly higher percentage of cases, people just kill themselves? Biology is not the cause of depression. There's no good sound basis for saying that it is. The serotonin theory of depression rests on corrupt studies done in foreign countries to evade scrutiny. I think we can let that go. Okay. Last thing to clarify, I'm talking to you as psychologists. Okay. You are honorary psychologists for the next 9 weeks, give or take, at those classes. I'm not talking to you as customers of psychology. If you're taking a thing for a condition and it's helping you, keep taking it and just ignore it. 1 or 2 of you, though, potentially could be a psychologist 7 or 8 years from now, or a psychiatrist. And I need you to have in the back of your mind that the epistemology behind these claims of biological origins, it just isn't that strong. Okay, so that's it for neurotransmitters for the time being, and we'll be back. I don't know how, but we'll be back soon to talk about brains.